So, the verse of the day today is, now is eternal life, that they may know you, the only true God, and Jesus Christ, whom you have sent. John 17:3. And we're going to start with respiratory burst. Respiratory burst, also called oxidative burst, involves the activation of the phagocyte NADPH oxidase complex, like neutrophils and monocytes, which utilize oxygen as a substrate, plays an important role in the immune response, which which leads to rapid release of reactive oxygen species. NADPH plays a role in both the creation and neutralization of reactive oxygen species. Myeloperoxidase contains a blue-green heme-containing pigment that drips butane its color. Phagocytes of patients with chronic granulomatous disease or CGD can utilize hydrogen peroxide generated by invading organisms and convert it to reactive oxygen species. Patients are at high risk for infection by catalase-positive species like Staph aureus and Aspergillus, capable of neutralizing their own hydrogen peroxide, leaving phagocytes without reactive oxygen species for fighting infections. Hyoscyanin of Pseudomonas aeruginosa generates reactive oxygen species to kill competing pathogens. Oxidative burst also leads to potassium influx, which releases lysosome enzymes. Lactoferrin is a protein found in circulatory fluids and neutrophils that inhibit microbial growth by iron chelation. Okay. All this is written kind of complicated. What they're saying, try to make it very simple. Um, what they're saying is how the body uh, kills off the bacteria, how phagocytosis happens. Okay, so this respiratory burst is how the body kills off bacteria by phagocytosis, and then using bleach in the very end to actually destroy the bacteria. See this right here. We're gonna start. We're gonna start step by step. The respiratory burst happens inside the phagolysosome of the neutrophil and macrophages after neutrophils eat the bacteria. The first step is phagocyte, which can be either neutrophil or macrophage, eats the bacteria. The bacteria is engulfed into a phagosome, which fuses with a lysosome. This forms a phagolysosome. So, inside the neutrophils, you have a phagosome and you have a lysosome. After the phagosome eats the bacteria, the phagosome fuses with the lysosome to form a phagolysosome, which is inside the neutrophil. Respiratory burst happens inside this phagolysosome. So this right here, this this pathway right here, this is the respiratory burst. This respiratory burst is happening inside of a neutrophil. And inside of the neutrophil, you have a phagolysosome. See this? This bubble right here is a phagolysosome. And inside of that is the respiratory burst. But if you look at the whole picture, this is like the entire thing would be a neutrophil. And inside the neutrophil, you have the phagolysosome. Inside the phagolysosome, you have the respiratory burst. Now we're going to go through the respiratory burst. Um, the bacteria is inside the phagolysosome, and this activates the respiratory burst. So first you encounter oxygen, then superoxide, and then hydrogen peroxide, and then bleach. So I think of this as O2, O2 minus, H2O2, bleach. O2, O2 minus, H2O2, bleach. And then in the middle of this, we have N, S, and then M. Only three enzymes. Okay? N, S, M. So Oxygen is converted to superoxide by NADPH oxidase. Superoxide is converted to hydrogen peroxide by superoxide dismutase. Hydrogen peroxide is converted to bleach by myeloperoxidase. That now you have the respiratory pathway um, down. Um, so eventually the bacteria is killed by the bleach, and that is how the body, uh, you know, phagocytoses and gets rid of bacteria. That's how all of our respiratory bursts are working properly. Okay, that's one of diseases. Now we're going to get to people who can't do the respiratory burst properly. Okay, so what you have to know is that H2O2, which is hydrogen peroxide, is important to kill the bacteria inside the neutrophil and macrophages. In CGD patients, which is chronic granulomatosis patients, NADPH oxidase is defective. This enzyme, the very first enzyme of the respiratory burst, is defective in CGD patients. So in CGD patients, NADPH oxidase is defective, so neutrophils cannot produce superoxide or hydrogen peroxide. So the bacteria cannot be killed because respiratory burst cannot happen. The bleach is not made in the CGD patients. So they have their bacteria is not killed. But some bacteria produce their own hydrogen peroxide, and then the body borrows that hydrogen peroxide to make bleach and kill off the bacteria. So even if you have NADPH oxidase deficiency, you'll be okay because the body will still kill off the bacteria. Okay, and that's in certain bacteria. The CGD patients will be okay because the bacteria have their own hydrogen peroxide. The bacteria have their own hydrogen peroxide, and then the body uses this hydrogen peroxide to go down this pathway and make bleach. So it starts from here and makes bleach. So who cares if the NADPH oxidase is working in CGD patients? But the problem is when you have catalase positive bacteria. So this is the problem with catalase positive bacteria. Okay. The catalase positive bacteria ruin the body's backup plan to kill the bacteria, which is for the body to use or borrow hydrogen peroxide from the bacteria to make bleach. This is because catalase positive bacteria break down the bacteria's own hydrogen peroxide using catalase. So this is because catalase positive bacteria break down the bacteria's own hydrogen peroxide using catalase. This is so the body cannot borrow the hydrogen peroxide from the bacteria to kill off the bacteria. So in CGD patients, we have catalase positive bacteria. The bacteria survive and can multiply inside the neutrophils and macrophages. I struggle so much with this catalase positive bacteria respiratory burst. So I'm just going to say the catalase positive part one more time. But catalase positive bacteria ruin the body's backup plan to kill bacteria, which is for body to use or borrow hydrogen peroxide from the bacteria to make bleach. This is because catalase-positive bacteria break down the bacteria's own hydrogen peroxide using catalase. This is so the body cannot borrow the hydrogen peroxide from the bacteria to kill off the bacteria. So in CGD patients who have catalase-positive bacteria, the bacteria survive and can multiply inside the neutrophils and macrophages. Okay, so these catalase-positive bacteria are not stupid; they're very, very smart because they know they have the hydrogen peroxide and they know that the body is going to take the hydrogen peroxide and use it to kill this bacteria. It's you know it, it's not self-destruct. The bacteria is not trying to self-destruct, right? So they also bring catalase, and catalase is what kills the hydrogen peroxide off. Yeah, the catalase kills off the hydrogen peroxide inside the bacteria, and now the body can't borrow the hydrogen peroxide, right? So the catalase positive bacteria survive like that and multiply and cause major issues. We'll get into it when we do the catalase positive bacteria. NBT. NBT is a dye added to the neutrophil in the lab to see if NADPH oxidase is working. NADPH oxidase makes superoxide, which reduces NBT to blue. If NADPH oxidase is not working, no superoxide will be formed and the NBT stays colorless. So a positive NBT test means respiratory burst is intact. The NADPH oxidase is working, producing superoxide that reduces the dye. Negative NBT test means NADPH oxidase is defective, so neutrophils cannot make superoxide, and the dye stays colorless. So in a positive NBT test, the test turns blue, and a negative NBT test, the dye stays colorless. That's it for respiratory burst. Next we have interferons. Interferons are interferon alpha, interferon beta, interferon gamma. Mechanism. A part of innate host defense, interferons interfere with both RNA and DNA viruses. Cells infected with a virus synthesize these glycoproteins, which act on local cells, priming them for viral defense by downregulating protein synthesis to resist potential viral replication and by upregulating MHC expression to facilitate recognition of infected cells. Also play a major role in activating anti-tumor immunity. Clinical use is for chronic hepatitis B virus, Kaposi sarcoma, hairy cell leukemia, condyloma acuminatum, renal cell carcinoma, malignant melanoma, multiple sclerosis, chronic granulomatosis disease, and adverse effects include flu-like symptoms, depression, neutropenia, myopathy, interferon-induced autoimmunity. So interferons interfere with viral replication. They are the body's antiviral warning system, and they also fight tumor. So, so interferons are important for anti-tumor immunity. So you have to know, like an example of this, of like interferon. They interfere with RNA and DNA viruses. They downregulate protein synthesis, so the body can't, like the virus can't replicate. And they upregulate MHC expression to kill off virus. Okay, so an example of this would be the liver, once infected with hepatitis B virus, tells other hepatocytes around it to stop protein production. So virus cannot overtake and use that hepatic protein for its own, for the virus's own good. Also, the liver tells other hepatocytes to increase MHC class 1 to fight hepatitis B virus. That's it for interferons. Next, we have cell surface proteins. So, cell surface proteins, for the T cells, you have T cell receptor, CD3, and CD28 all on top of the T cell. T cell receptor on the T cell binds antigen on MHC class 1 or 2, which is on the APC. And every T cell has CD3. CD28, which is on the T cell, binds B7, which is on the APC. Helper T cells have CD4 and CD40 ligand and CXCR4 and CCR5 on top of the helper T cell. So CD40 ligand on the helper T cell binds to CD40 receptor on the B cell. Remember we talked about this? CD40 ligand is always on the T cell, specifically helper T cell, and that CD40 ligand binds to CD40 receptor on the B cell. Next, CXCR4 and CCR5 are both on the helper T cell, and this binds to HIV. Next, we have cytotoxic T cells. CD8 is on the cytotoxic T cell, and CD8 binds to MHC class 1 on the APC. Yeah, CD8 is a receptor on the surface of the cytotoxic T cell, and that binds to MHC class 1 on the APC. The regulatory T cells have CD4 and CD25. CD4 and CD25 receptors are on the surface of regulatory T cells. CD4 on T regulatory cell binds MHC class 2 on the APCs. CD25 on the T regulatory cells binds IL-2, which grows T regulatory cells. B cells. B cells have immunoglobulin on top of it. B cells also have CD19 and CD20 and CD21 and CD40. And B cells also have MHC class 2 and B7 on top of it. So immunoglobulin exists by itself on the B cell surface and eventually binds antigen. CD21 is a receptor for Epstein-Barr virus. Okay, so CD21 on the B cell CD21 on the B cell surface Yeah, CD21 is on the B cell surface, and then Epstein-Barr virus goes and binds to the CD21 on the B cell. Okay. MHC class 2 and B7, all APCs will have B7 and MHC2, and a B cell is a type of APC. That's why MHC class 2 and B7 are written here. Next we have macrophages, CD14 and the CD40 and CCR5, MHC class 2 and B7 are all on the macrophage. FC and C3b receptor. So FC and C3b receptor are on the macrophages also. So CD14 is on the macrophage and binds LPS, which is a type of PAM, from gram-negative bacteria. This activates TLR4, leading to innate immune activation. MHC class 2 and B7 all okay. MHC class 2 and B7, all APCs will have B7 and MHC class 2. Macrophages have C3b and FC receptors on them. FC portion of IgG binds FC receptor on the macrophage. C3b binds to C3b receptor on macrophage for opsonization. So CD14 on the macrophage binds LPS, which is a type of PAM, from gram-negative bacteria. So LPS is on the gram-negative bacteria, and CD14 goes and binds that LPS. And this activates TLR4, leading to innate immune activation. So LPS is a component of the outer membrane of gram-negative bacteria. Gram-positive bacteria have something known as lipoteichoic acids, but that is, something else. We're only talking about gram-negative bacteria here when we talk about LPS, which is lipopolysaccharide. It's on the outer membrane of the gram-negative bacteria. CD40 and TCR5 are also on the macrophage. FC and C3b receptors are also on the macrophage. Macrophages have C3b and FC receptors on them. FC portion of IgG binds FC receptor on the macrophage. FC portion of IgG binds FC receptor on macrophage. C3b binds to C3b receptor on the macrophage for opsonization. Okay, next. Uh, Natural killer cells. Natural killer cells have CD16 on them. This binds Fc region of IgG. Natural killer cells also have CD56 on them. Yeah, so natural killer cells have CD16 and CD56 on them. CD16 binds Fc region of IgG. Binds Fc region of IgG. Remember we talked about this? CD16 on the natural killer cells binds Fc region of IgG. CD56 on the natural killer cell is another marker for natural killer cells. Next, we have hematopoietic stem cells. They all have CD34 as a marker. Uh, Hematopoietic stem cells makes all blood cells in the body, like B lymphocyte, T lymphocyte, natural killer cells, antigen-presenting cells, platelets, RBCs, neutrophils, and eosinophils. Okay, and we're done with cell surface proteins. We have anergy. This is a state during which a cell cannot become activated by exposure to its antigen. T and B cells have become anergic when exposed to their antigen without co-stimulatory signal. Another mechanism of self-tolerance. So what they're saying is T and B cells become anergic if they don't have a co-stimulatory signal. So they need the second signal. T and B cells need the second signal to become fully active. If not, they just become non-energetic is how I think of it. They can't become activated. Um, they can't become activated even if they are exposed to a bacteria. They need both signals to work. This is another example of self-tolerance. Self-tolerance is the ability to recognize your own cells and not attack them. So here, the the body does not attack its own B and T cells, even though the B and T cells are non-functional. That's how the body, um, you know, doesn't destroy its own B and T cells, even like though they are silent. They just keep them there. They're alive. They're not dead, so they keep them there. It just doesn't launch an immune attack against them and destroy them. Because if you did that to all the B and T cells, um, that are not active, they're not actively fighting for you, you're going to destroy your B and T cells. And this is what causes autoimmune disease. Okay, next we have passive immunity and active immunity. Um, Means of acquisition. So passive immunity, it's when you receive pre-formed antibodies. The onset of passive immunity is rapid. Duration of passive immunity is a short span of antibodies. So the body will clear the antibodies in 3 weeks. That's what that's saying. So half-life equals 3 weeks means that the body will clear half of the antibodies in 3 weeks, not the whole thing. Before I said it will clear the antibodies in 3 weeks. No, it will only clear half of the antibodies in 3 weeks for passive immunity. Um, it will only clear half of the injected antibodies in 3 weeks. And examples of passive immunity would be IgA in breast milk, maternal IgG crossing the placenta, antitoxin, humanized monoclonal antibody. Notes: after exposure to tetanus toxin, botulism toxin, hepatitis B virus, varicella, rabies virus, or diphtheria toxin, unvaccinated patients are given preformed antibodies to be healed very rapidly before dying. So passive immunity is only done by injection or blood or milk from mom. So receiving preformed antibodies means uh, like your mom made it before uh, for the baby. Like IgG. So your mother made that for you before you're born. IgA in breast milk. Colostrum has increased like the highest amount of IgA. And antitoxin has antibodies to snake venom. Antitoxin and humanized monoclonal antibody both have antibodies to snake venom. So passive immunity is when the body is given antibodies that was made by someone or something else, but not made by the person's own immune system. However, active immunity is when the body makes its own antibody. Means of acquisition for active immunity is exposure to exogenous antigens. The onset is slow. Duration of active immunity is long-lasting protection because B cells can become plasma cells, immunoglobulins, or memory cells. Okay. Uh, so because in active immunity, body is exposed to an antigen through infection or vaccination, uh, which activates the B cells. Some of these B cells become plasma cells, which make antibodies now. Other B cells become memory B cells that stay in the body to respond faster and stronger if that same bacteria comes into the body again. That's why we write long-lasting protection and memory next to it. Examples: natural infection, vaccines, and toxoid. Um, Notes: combined passive and active immunization can be given for hepatitis B or rabies exposure. Combined and passive and active immunizations given because rabies has a 100% mortality rate. Okay, so for rabies, give combined passive and active immunization. So if you don't get vaccinated for rabies, once a infected dog or bat bites you, you will die. You must have vaccination for that. Um, and the vaccination that's given is combined passive and active immunization. And the rabies vaccine is you only give killed rabies vaccine in the injection. So combined passive and active immunization uh, for hepatitis B and rabies exposure. Okay, next. So for vaccination, um, we have vaccination induces an active immune response, humoral and or cellular, to specific pathogens. So it can either vaccination can activate either Th1 or Th2. Th1 would be cellular, Th2 would be humoral. Okay, a live attenuated vaccine. This is when microorganisms loses its pathogenicity, but retains capacity for transient growth within inoculated host. Induces cellular and humoral responses. MMR and varicella vaccines can be given to HIV-positive patients without evidence of immunity if CD4 cell count is over or equal to 200 cells per mm. Pros and cons of live attenuated vaccine. Include induces strong, often lifelong immunity. Cons may revert to a virulent form, often contraindicated in pregnancy and immunodeficiency. Examples include adenovirus, non-attenuated, given to military recruits, typhoid, TY218 oral, polio, Sabin, varicella, chickenpox, smallpox, BCG, yellow fever, influenza, which is intranasal, MMR, rotavirus. Attenuated means decreased power of virus. So here it is, live attenuated vaccine is still a live vaccine, but the strength of this live vaccine is decreased. It says microorganism loses its pathogenicity but retains capacity of transient growth within inoculated host. So basically, the bacteria is not harmful anymore to the patient that's being uh, vaccinated, but the bacteria still grows enough to cause a response. Now, it says induces cellular and humoral response. This means that live attenuated vaccine, increase in macrophages um, because it induces cellular response, and the live attenuated vaccine also increases immunoglobulin and memory cells by CD4 positive T cell because the live attenuated vaccine also provokes a humoral response. A humoral response, it means that it um, increases the immunoglobulins and memory cells in the body. And here it says you can give live attenuated vaccine to HIV patients if their CD4 cell count is over 200 cells. So this means HIV patients can get MMR and varicella vaccine if CD4 count is over 200, if the patient has no evidence of immunity, which means you can give them MMR and varicella vaccine if their CD4 count is over 200, if they have no vaccines, if they have no MMR or varicella vaccine history, and or no antibodies to these MMR or varicella. So MMR and varicella vaccines are both live vaccines and are generally avoided in immunocompromised people, but in HIV-positive patients with a CD4 count above 200, their immune system can handle this live vaccine. So HIV patient, if their CD4 count is over 200 and they don't have any antibodies to MMR or varicella, and if they don't have any record of MMR or of being given MMR or varicella, you must give them the MMR and varicella vaccine, but only if their CD4 count is over 200, equal to or over 200 cells. And the reason that they have lifelong immunity, the pros of life attenuated vaccine, the pros are they often have lifelong immunity. This is due to memory cells, which come from B cells. So, and then here, adenovirus, it says non-attenuated, given to military recruits. So, for adenovirus, the virus is stronger because, well, adenovirus, it says non-attenuated. So basically that adenovirus is not weakened. The virus is not weakened. It's stronger, right? And that is given to military recruits. Because we're talking about live attenuated vaccine, which is MMR and varicella. But when we're talking about adenovirus, that adenovirus is not weakened. And that is what's given to military recruits. Now, influenza, influenza is a live attenuated intranasal spray given to healthy, non-pregnant patients ages 2 to 49 years of age. So, influenza is a live attenuated intranasal spray given to healthy, non-pregnant patients ages 2 to 49 years of age. Okay, that's fine. Got it. So killed or inactivated vaccine, the pathogen is inactivated by heat or chemicals, maintaining epitope structure on surface antigens is important for immune response, mainly induces a humoral response. Pros, safer than live vaccines. Cons, weaker immune response. Booster shots usually required. Um, the examples of killed or inactivated vaccine would be hepatitis A, typhoid, VI polysaccharide, intramuscular, and then rabies, influenza, polio, which is the Salk vaccine. Okay. Killed or inactivated vaccine means the same thing. And it says maintaining epitope structure on surface antigens is important for immune response. Okay, maintaining epitope structure on surface antigens is important for immune response. This means the structure of antigen is retained so the B cell can bind CD4 positive T cell. The structure of the antigen is retained so B cell can bind CD4 positive T cell. Pros, it's safer than live vaccines because, killed is better. They won't harm you. But the cons is it is there is a weaker immune response. And here you need booster shots. Examples of this of the killed vaccine would be hepatitis A and typhoid. This VI polysaccharide is found in the typhoid vaccine, and it's given intramuscularly. Rabies is always killed vaccine. Salk is the name of a polio vaccine. Next we have subunit. Subunit includes only the antigens that best stimulate the immune response. So this means that subunit vaccines are vaccines which are made from antigens taken from the virus. These antigens are the main antigens that give you infection. So if we develop antibodies against these antigens, by getting this vaccine, the virus cannot infect us. So this subunit vaccine is a vaccine made from antigens sitting on top of virus cell membrane. The pros is, lower chance adverse reactions because it's only one subunit. It's not a whole bacteria, it's only a piece of bacteria, and that's enough to evoke a immunologic response once you're vaccinated with this subunit of bacteria. It's not a bacteria, it's a virus. So, hepatitis B virus. How hepatitis B enters the body is through hepatitis B surface. So, how the hepatitis B enters the body is through hepatitis B surface antigen. So the vaccine was made that, okay, how, yeah, so the vaccine that was made that will make the antibody against HBsAg, So the antibody will be bound to HBsAg. So what that means is HBsAg is the hepatitis B surface antigen that is a viral envelope protein found on the surface of hepatitis B virus, and it is through this HBs antigen that hepatitis B virus enters the liver. Now what happens when you get vaccinated with hepatitis B is that the hepatitis B vaccine, so hepatitis B vaccine contains HBs antigen, and when injected, the body makes antibodies against HBs antigen, and these are called anti-HBs. Anti-HBs antibodies neutralize hepatitis B virus by binding to hepatitis no, anti-HBs antibodies neutralize hepatitis B virus by binding to HBs antigen, preventing the hepatitis B virus from entering the liver cells. I'll say it again. HBs antigen is the hepatitis B surface antigen that is a viral envelope protein found on the surface of hepatitis B virus, and it is through this HBs antigen hepatitis B virus enters the liver. What hepatitis B vaccine does? Hepatitis B vaccine contains HBs antigen, and when injected, the body makes antibodies against HBs antigen, and these are called anti-HBs. Anti-HBs antibodies neutralize the hepatitis B virus by binding to HBs antigen, preventing the hepatitis B virus from entering the liver cells. So that's how the vaccine uh, stops hepatitis B. Because the anti-HBs antibodies neutralize hepatitis B virus by binding to HBs antigen. So anti-HBs antibodies bind to HBs antigen, and this stops the hepatitis B from infecting the liver cells. Side note, if a patient has anti-HBs but no anti-HBc, that means they were vaccinated and not exposed to the actual hepatitis B virus. And that is, again, if the patient has anti-HBs but no anti-HBc. Okay, next, toxoid. Denatured bacterial toxin with an intact receptor binding site stimulates the immune system to make antibodies without potential for causing disease. Pros: protects against the bacterial toxins. Cons: antitoxin levels decrease with time, may require a booster. Examples include Clostridium tetani, Corynebacterium diphtheria. So toxoid, you have denatured bacterial toxin with an intact receptor binding site. Stimulates the immune system to make antibodies without potential for causing disease. So when they say denatured bacterial toxin with an intact receptor binding site, they're saying this is a heated up toxin, so toxins cannot work. These toxins are injected into the blood, so the body creates antibodies to the toxins. And the intact receptor binding site it means that the structure of the toxin is retained, so antibodies can still be made. So basically, this bacterial toxin is heated up so the toxins cannot harm the patient, but the antibodies will still be made. And then denatured bacterial toxin, the structure of the toxin is retained, so antibodies can still be made. Pros, protects against the bacterial toxins. Cons, antitoxin levels decrease with time, may require booster. So what that means, the con is antitoxin levels decrease with time, may require a booster. Okay, an antibody. So this antitoxin is a type of antibody. An antibody, like an antitoxin, binds to a bacteria and blocks its bad effects. So the toxin can no longer enter and damage those cells. So the con of toxoid vaccine is that these antitoxin levels can go down over time. So then if the antitoxin level goes down, there's nothing to bind to the bacteria and stop its bad effects. A toxoid is a vaccine that's made from the toxin that is secreted from the virus. The toxoid vaccine is made from toxin that is secreted from the virus. An example of this would be Clostridium tetani and Corynebacterium diphtheria. So basically, Corynebacterium diphtheria secretes secretes a toxin, and you make the toxoid from that. So except Corynebacterium diphtheria and Clostridium tetani are both bacteria. Not virus. So I like to correct myself. Um, Toxoid is not used for viruses at all. It's only used for bacteria. So toxoid is a vaccine that is made from toxin that is secreted from the bacteria. An example of this would be diphtheria. Okay, next. Hypersensitivity hypersensitivity types. There's 4 types. Hypersensitivity types, there are 4 types. Anaphylactic and atopic, which is type 1, antibody mediated, which is type 2, and then immune complex, which is type 3, and delayed hypersensitivity, which is also known as cell mediated, and this is a type 4 reaction. So what does cell mediated mean? Th1. What is Th1? Increase in macrophages. So the one that has to do with macrophages. An increase in macrophages, that is the delayed type of hypersensitivity. Now, types 1, 2, and 3 are all antibody mediated. Type 4 is the only one that has to do with macrophages. Extremely high yield for step 1, probably step 2 also. Let me know if you want me to make videos for step 2 in the comments, or continue videos with step 1. Okay? Alright, thank you. Type 1 hypersensitivity, anaphylactic and atopic. There's 2 phases. Immediate, which just happens in minutes. Antigen crosslinks, pre-formed IgE on pre-sensitized mast cells. and this leads to immediate degranulation, which leads to release of histamine, a vasoactive amine, and tryptase, a marker of mast cell activation. The late type happens in hours. There are chemokines, which attract inflammatory cells like eosinophils and other mediators like leukotrienes from mast cells, and this leads to inflammation and tissue damage. The test is a skin test or blood test, which is ELISA, for allergen-specific IgE. Example would be anaphylaxis, like food, drug, or bee sting allergies, or allergic asthma.